So welcome to the first video on the physiology series. Um, if you look in the links below, there is a link to this picture as well as a link to the notes associated. So let's get started. So as we know, you have your cell membrane on the outside, and it could also be called, so your cell membrane can be titled many things. It could be called a plasmalemma, or the plasma membrane. In the case of this, we have a bilayered membrane. And one thing you should always know is that potassium concentration is high on the inside, and on the outside, sodium concentration is higher. So that would mean that if we're looking at the inside, we're going to see a lower sodium concentration and a higher potassium concentration. And in later videos, we're going to go over what those exact numbers are and stuff like that. And so this little thing that I've drawn right here, this is a pump that's going to control this concentration gradient. And so basically what the function of this membrane is, it's to basically control what comes in and out of the cell. So it's going to basically say, okay, what nutrients do I need right now? Let me make sure they're inside the cell versus not. Anytime there's waste products, it's going to remove those waste products, and basically it wants to always control the composition of the cell to ensure that everything's going to be okay. Because the last thing you want is a high concentration of sodium, for instance, on the inside, creating more problems for the cell, because that could lead to the cell shriveling up. While if there's a low concentration, not shriveling up, but kind of blowing up and lysing, while if there's a too low of a concentration of sodium and a lot of sodium on the outside, you can have the cell shrivel up. Now if we were to look at the cell membrane and kind of blow it up, we can see what it's made of. So it's basically made of lipids, lipids, proteins, and carbs. In terms of the carbohydrates, it's going to be in the lowest amount. Now, if we were to look at the lipids, the lipid composition is going to be phospholipids. Then we're going to also have cholesterol, and then glycolipids. And then the percentages, you're going to have more phospholipids than anything else. So it's about 70% phospholipids. Cholesterol, it's about 20%. And 10% is the glycolipids. And always remember, you know how everybody says cholesterol is bad for you? Well, your body needs cholesterol to survive. Just keep in mind which one's the good versus the bad. And so these concentrations are going to make sure that, or because it's a lipid, what that means is that it's a fat. And because we have a fat here, things that can cross are going to be kind of, remember general chemistry, like dissolves like. It's the same idea. If we have things that are oily, it's going to be able to pass through because it's got that same level of charge. But basically, the lipids are going to decrease the possibility of anything passing through except for gases, specifically O2 and CO2. Now, if we're looking at the proteins, on the other hand, the proteins, they're going to define the functional characteristics. So they're going to define the shape. It's also going to be involved in transport. So you've got your structural transporter, your structural support, your transporter enzymes, your hormone receptors, your antigens, your ions and water channels. So your proteins are going to be more important as how you're going to transport things from in and out of the cell. How are you going to send signals inside the cell? So for instance, if you need this specific cell to have a different function or a specified function, you want to have a signal come up to the receptor, whatever it may be, whatever type of receptor it is, and that receptor is going to send a signal inside, and that signal is going to go to the nucleus and say, okay, time to make that protein, or time to go through transcription and translation. Now if we look at if we were to take one block right here of the cell membrane and look specifically at what the structure looks like, we're going to get something like this. And what this is we've got a hydrophilic head, and the tail here is hydrophobic. And so what this means is the tail is the fatty acid end, while the head isn't. It's going to be the more polar part of it. And one way to kind of think of this is you can almost think of it as Scotchgard. When you place, you know how like there's those commercials with people spilling wine on their carpet. Well, this is the same thing. The hydrophilic head is going to keep the wine from touching the carpet. And the same thing happens in the body. So the hydrophilic head is going to keep this liquid portion outside of the cell, so it doesn't come, so the stuff on the outside, the liquid on the outside, and all the gunk that's along with it can't come inside the cell because we have this hydrophobic end. And the hydrophilic, they're going to want to go next to each other. And basically the tails are going to want to line up next to each other because they like each other. Think of magnets almost. And so we're going to have one set of hydrophilics facing the extracellular space and another set facing the intracellular, because we have what's called a lipid bilayer. And it's going to kind of look like this. And so this kind of goes into the fluid mosaic model, where it shows this lipid bilayer plus there's integral proteins, which we're going to get into later, and transmembrane proteins that are going to be able to allow for communication between this lipid bilayer. The other thing to know is how do these amphipathic molecules move around? So a big, Jeopardy word is amphipathic, meaning it has two functions, so a hydrophilic and a hydrophobic group. So one way that this lipid can move around is it can do rotation, so it can rotate along an axis. The other is we can have the fatty acid groups bend, or they can undergo lateral diffusion. So what that means is, if this is within the lipid bilayer, this individual unit has the ability to move to another space. And on rare occasions, it can move up. So, you got to forgive me for the way I drew it, but if we have the lipid bilayer here, this red one I've drawn, what it's done is it's actually done this kind of crossing over movement and gone up the lipid bilayer. This happens very rarely, and it's not really something that's always seen. So if you guys have any comments or questions, please leave them below. If you like the video, give it a thumbs up, and if you like the channel, please subscribe. Thank you.